need everybody all you got this, bro. You got this. Demerit cards should be out. Please get your demerit cards out. Good. Excellent. Here's the deal. Your do first is on the board. You picked up a piece of paper that says do first at the top. You should answer 1 through 3 in complete sentences on your do first paper. I am collecting these. Complete sentences. There should be absolutely no talking. Gracie, who are we using? Perfect. You have about 2 minutes remaining. Once you're done, check your answers by looking around the room. Remember, 9th graders use their resources. Check around the room. Lots of complete sentences. Make sure your sentences. Good complete sentences. Good practice for our free response coming up next week on the common assessment. Less than a minute. It up in a few seconds. All right, couple of things. So you guys have already seen the beads. I went to Mardi Gras this weekend at Galveston, and they tossed out a ridiculous amount of Mardi Gras beads. And at first I was like, oh, that's a massive waste of plastic. I don't want any beads. And then my partner was like, hey, give them to your kids. And I was like, give me beads! So if you saw earlier, I had like this whole thing filled with beads and I've given them all away. You each have one of your own. And you have the opportunity to win some pretty cool ones. There's bananas on this one. By volunteering today. So, here's the deal. If I either call on you randomly, you're giving us your best so you can earn one of these. Paying super close attention so you know you've got the right answer, you know you're going to get a banana. Other things. So remember we've got those 5 strikes. Those count now. Towards going back to the regular seating arrangement, you guys are going to be fine. But remember, make sure that side conversations, no side conversations. We're not calling out or anything like that. All right, so what we're going to do is go through each of the do first questions. And for these, I'm going to randomly call on someone. So Isaiah, for the chance at a bead, tell us, during which phases of the cell cycle does the cell grow? So I need everyone tracking Isaiah. He's going to stand with pride and tell you his answer in a complete sentence.
This cell grows during growth 1 and growth 2?
Yeah. Sorry, Sean. I almost killed Sean. During which phases of the cell cycle does the cell replicate its DNA? Bam, Marina's not here. One more time. Bam, young man, tell us.
The DNA is replicated during the synthesis stage in the cell cycle.
Bam, 2 purple ones for you. Last one. During which phases of the cell cycle does the cell divide, Aaron?
The cell divides during the mitosis cycle.
All right. Excellent. Does everybody have those same answers?
Yes.
Awesome. If you didn't, make sure that you changed them. You got them all right. I am going to collect that due first. So we talked last week about the cell cycle. We talked about how this is a series of events that causes a cell to make an exact copy of itself. Exact copy of itself. This is also known as the cell cycle, the seasons of a cell's life. So can I have a volunteer for a bead? Read me the definition of mitosis. Maya, go for it.
Mitosis, subdivision in eukaryotic cells that result in 2 identical daughter cells. Mitosis is responsible for making somatic cells.
Can you read that part in red for us just one more time, nice and loud?
2 identical daughter cells.
Identical. What?
Identical.
Identical daughter cells. Thank you, Maya. Yes.
What do you mean by daughter cells?
So you start out with one cell, that's like the parent cell, and then you end up with 2 cells that are identical after that. They call it daughter cells. You start with one parent, and then you end up with 2 little babies. They're daughters. But they're actually clones. They're actually supposed to be clones. Yes?
So is there a time where one of them has a mutation and it's different from the other?
That can happen. There are also, remember we talked about non-disjunction errors a long time ago, where you end up with more copies of a chromosome in a cell than you're supposed to, like Down syndrome, trisomy 21. Remember that? That can happen as well. There's a couple of errors that can happen with this. But if it happens properly, it should be identical. So if a cell starts with 4 chromosomes, and it wants to make an identical copy, how much should those ending cells have?
2.
Identical. You start with 4, you should end with 4. Excellent. That's kind of confusing. That's a hard topic to kind of conceptualize, but what we go through today will help you understand that. Who wants to read us somatic cells? Sean.
Somatic cells. Every cell in the body except
Gametes.
gametes, reproductive cells.
Can you tell me, Sean, for this long purple one, what the reproductive cells are? There's 2 of them. What are the 2 reproductive cells boys and girls have?
X and Y. Or is it something?
No, those are chromosomes, the cells. What are the sex cells of girls and boys? Someone want to help them out? Alexis, we haven't heard from you yet. Go ahead.
Sperm and eggs.
Sperm and eggs. Excellent. So we're going to do a quick big picture. If a human has 46 chromosomes in their cells, and their cell undergoes mitosis, how many chromosomes will the resulting 2 cells have? I want you to think about this. Write down your answer on the back of your do first. Again, I am collecting this, so make sure you're writing everything down. If you start with 46 and you undergo mitosis, an exact, identical copy each cell have in the end. Each cell, not combined. Just one cell. How much should just one cell have? That's a good question. Okay, hang on 1 second to give you a pair. Does everybody have an answer answer down. Raise your hand if you need more time. Okay, go ahead and pair up with the people around you and tell them what you think. Go ahead. That's what we started. It's supposed to be the same exact amount. So you are right. You guys have one. What do you guys have? Aha! Okay. So who's right? If you start with 46 and you want to make identical daughter cells, how much does each daughter cell have? Identical. No, that would get you combining both cells. But we're just talking about each cell should have
Also,
the
like,
same.
so instead of saying how much did each have, how many close.
Sure, yeah. All right, let's bring it back, and I'm going to randomly call on somebody to answer for us. Maya, what did you get? What did you discuss in your group?
Me and my group discussed that at the end, the resulting cells will have 46 cells.
46 cells, and why?
Because they're identical, so they just replicate.
Yeah, we want identical daughter cells. And let me draw this out for you. During interphase, if you can't remember, look over there at the chart on the board. What happens during synthesis? Read that out loud for me. DNA replicates. If we've got 46 to begin with, we replicate that amount of DNA. What's 46 times 2? 92. Those 92 chromosomes are going to get split up during mitosis between the two, and you end up with, what do we end up with? 46 in each. So we start with 46, we replicate in synthesis during interphase, and we end up with 46. Excellent. All right. So now we're going to go through the phases of mitosis. So you picked up, as you came in, you picked up one of these. You're going to be filling this in as we go. This will help you with our lab that we're starting today and finishing tomorrow. Interphase, this is before mitosis. We've talked about this at length. This is where the cell grows and makes another copy of DNA. You've already got that written down, right?
Yeah.
This is the image I want you to think of. Everybody do this. Everybody's like, oh my God, how'd you take this picture? Somebody took it for me. I'm not that talented. I can't take pictures with like a mouth holding the camera. This is interphase. This is what I want you to think of when we talk about interphase. Prophase. Please write down the underlined portions. Prophase is the longest phase of mitosis. This is where the cell's chromosomes become visible. They look kind of like spaghetti. And that nuclear membrane breaks down. Right, as quickly as you can, so you've got lots of time on your lab. College prep. Chromosomes are visible, kind of looks like spaghetti, and the nuclear membrane breaks down. So I want you to think of prophase, which starts with a P, as like the preparation phase. Prophase, prepare. If you want to write that at the top, I see Aaron writing that above prophase. Prophase is like the preparation stage, prepare. This is the hand gesture I want you to think of when you think of prepare, prophase. So what was interphase again? Show me. Interphase. Prophase. Y'all look at my hands. Stay on one. Metaphase. That's where the chromosomes line up in the middle or at the equator. Chromosomes line up at the equator of the cell. And when you're you're done writing that down, the way I want you to remember this, metaphase, M for meet or middle. Meet or middle. The hand gesture I want you to think of for this is this, together. I promise my fingers don't look that fat in real life. I stretched out the photo. I swear. Or I was bloated yesterday. Okay, got that one? What was interphase again?
Like this.
Let me see. Prophase? And metaphase. Excellent. Remember that order. Remember that order. Hint, hint. Anaphase. This is where the chromosomes pull apart. The chromosomes pull apart. What do you think I want you to remember for anaphase? Apart. Apart. Aaron's already got the hand motion down. And I haven't even shown it to you yet. Chromosomes pull apart. What was our hand gesture for metaphase? Metaphase.
Oh, I think they need this.
Meet. What do you think apart is going to be?
They go
Like
like this.
anaphase. All right. So that's the hand gesture. I want you to know you're pulling them apart. That's anaphase. Got it? Shut up on my fingers. Okay, telophase. The last one I want you to fill in because we know cytokinesis. Telophase. That's where the nuclear membranes reform and the cell begins to tear. T, tear. I want you to remember tear for telophase. Tear for telophase. And then cytokinesis, we know that one. That's just where the cell splits. Wha-cha! Got it?
Yeah.
For this one, this is the hand gesture for telophase. We were at metaphase, anaphase, telophase. What do you think we're going to do for cytokinesis? Oh, I kind of like that. The cell doesn't beat itself up. What did we do last time for cytokinesis? Yeah, wha-cha! I even put it in it. Ready? So, let's go through this together. What was interphase? Interphase? Prophase. Metaphase. They meet in the middle. Meet in the middle. Anaphase. They get pulled apart. Telophase. Now they're in their own little nuclei. They've got their own nuclei. They're becoming to get a little messy again. And then cytokinesis. So does anyone with the big bananas want to do that all for me without looking at their notes? Including the names and the correct hand gestures. Mario's the only brave one. I have faith in you. Mario, go ahead, give it a shot. I know you want the bananas. Go ahead and stand in the front of the class for me.
Yes, please, Mario.
Watch him closely now. Don't help him.
It's
Okay.
interphase.
Interphase.
Prophase. No wait.
Yeah.
Prophase.
Prophase. There we go.
Metaphase.
Meta, not metal. I like that though. Metaphase.
Metaphase. Ooh. Have a clue?
This is where they pull apart.
Oh.
Part of phase. I like it, but that's not it. Try again.
I'm thinking of possible. Anaphase.
Uh-huh.
Okay.
And
There was telophase.
and
cytokinesis with chop.
what shot?
Yes!
Well done, Mario. Well done. All right. He worked hard for those bananas, y'all. Okay, so how the heck are we supposed to remember this order? There's a bunch of stuff in there. Interphase, prophase, metaphase, anaphase, telophase, cytokinesis.
What?
So that's a lot to remember. How are you supposed to remember that order? Well, if you're like Mario and you work really hard, you might be able to remember the steps of the hand gestures. You could also remember IP, NATC. Who's going to remember that? that. My personal favorite that I recently learned, I peed on the mat. See? So I for interphase, P for prophase, I peed on the M for metaphase, A for anaphase, T for telophase, and C for cytokinesis. Got it? I peed on the mat. See? You have to show them too. You can't forget to show the people that you peed on the mat. Got it? I peed on the mat. So I would love it if you haven't already to write that somewhere on that sheet of paper. I peed on the mat. See? That will help you remember when you take this information and try to translate it to your common assessment. Yes. What? My bad. I peed on the mat. I peed on the mat. See? No, I didn't come up with this. I'm not that brilliant. You got it? I peed on the mat, see? Everyone have that down? Yes. Good to go. All right. So here's a video that I want you to watch of a real cell in the process of undergoing mitosis. We can leave the lights on because you can see.
Differential interference contrast microscopy is used here to visualize mitotic events in a lung cell grown in tissue culture. Individual chromosomes become visible as the replicated chromatin starts to condense.
This is prophase. Prophase.
The two chromatids in each chromosome remain paired as the chromosomes become aligned on the metaphase plate.
They're aligned, so we're at metaphase.
The chromatids then separate and get pulled by the mitotic spindle into the two nascent daughter cells.
They're pulled apart, so anaphase.
The chromatin decondenses as the two new nuclei form, and cytokinesis continues to constrict the remaining cytoplasmic bridge until the two daughter cells become separated.
So that's telophase, where they're starting to tear, and cytokinesis, when they break apart. Excellent. Here is another video where there's no sound. So we're going to go through this one more time. See if you can identify the pieces. Here's prophase, looks like some spaghetti. Looks like they're on the move. They're lining up. What's that called? Metaphase. Metaphase. Now they're being pulled apart. What's that called?
Anaphase.
Anaphase. And you see the cells start to constrict in the center. What's that? Telophase. Just something interesting. Something interesting here towards the end, as they're starting to tear. Can you see that line form in the center?
Yeah.
That's the new plasma membrane boundary. You can't just leave it, naked on one side, right? You have to fill that in with the plasma membrane, have the bouncer of the club be complete all the way around. That's the new plasma membrane forming. Super cool. That is a real cell undergoing mitosis. So what we're going to do, again on the back of your do first, we're going to fill these in. Get some practice because you'll have to do this on your common assessment for sure. I'm going to do number 1 through 4 with you. Number 1, it looks like they're meeting at the center. What do we call that? Metaphase. Metaphase. You can just write M to save time. 2, it looks like they're getting prepared. We've got some squiggles. We can see the chromosomes. And that nucleus is beginning to break down. Shout it out. What is it?
Prophase.
That's prophase. P for prophase. Number 3, it looks like they've already separated. What do we call that? Oh. Cytokinesis. That one's C. And number 4, what do we think that is? We don't see anything going on yet. Looks like it's still maybe in the interphase part. There we go, interphase. 5 through 13, I want you to label those on your own, and I'm going to come around and check. Shouldn't take you any more than about a minute. There's a couple of them. Mine's already done. So is Gracie. Take another 15 seconds to wrap it up if you haven't finished yet. All right, what's 5? Shout it out. Interphase. What's 6? Interphase. 7? Yeah, we see those chromosomes being pulled apart. So that's anaphase. 8? Interphase. 9? Anaphase. That one's anaphase. Again, chromosomes being pulled apart. 10, whatever that number says. 10? Interphase. That's interphase. We've got a meeting in the middle at 11. What is that? Interphase. 12. Looks like they've already maybe split. I don't know. We can call that telophase. Telophase or cytokinesis. It's Karen. We got that. All right. And 13? Interphase. Excellent. So now we're going to get started on our onion root tip lab. Your group's mission. You are scientists trying to understand the cell cycle and how cells reproduce. You are going to find an envelope full of cut up onion root cells. I was recycling paper, so this is your envelope. Some of you guys helped me cut these up, thank you. You're gonna find those cells, and this is what your group is gonna do. Once I have everyone's eyes, I'll explain what you need to do and then let you go. You ready? Your group is gonna look at the pictures of the real cells. You're going to organize them into 5 groups based on how they look. Put them all into a group based on what they look like. This one looks like they're being pulled apart. What do we think this is? Anaphase. Yeah, so we would put that in our little anaphase pile. We're going to order the groups. I peed on the mat, see? Order the groups and then describe and name them. And you're going to go through this lab manual here. Everyone has a copy. You will share that copy for your table, and I'm going to collect it at the end of class, and then we're going to finish up on it tomorrow. Make sure you're moving quickly. The expectations, I want you to trade your lab manual. Not done yet. I know you're excited. Eyes up here. You're going to trade the lab manual every question so that everyone's being held accountable. Make sure that everyone's working. If you reach a stop sign, raise your hand. Make sure you're on the right track. Hold each other accountable to excellence. Don't let anybody slack off. And I want you to answer every question to the best of your ability. If you're not sure, ask for help. And then we're going to take an exit ticket during the last 5 minutes of class. So make sure you're working quickly. Go ahead and get started. Let me know if you need any help. Yeah. Just one. You got your envelope? All right, bring them out. See what you got. Yeah, make yourself some room. Yeah. Sort them as quickly as you can. Sort them quickly. Sort them quickly. Yeah, all of them. Yeah, it's awesome. Those are onion roots.
It's mine.
That's yeah. Move them over and separate them as quickly as you can. Check how to do this. Yeah. So what do you think? Show me one that you think is an interphase. Yeah, they have dark circles. They haven't really done anything yet. Show me one that you think is in prophase. You can kind of see the chromosomes. Prophase. And you can use this as a guide. You can it kind of looks like spaghetti. Find me one of those. Yeah, maybe that one. This one would be good too. Kind of looks like a bowl of spaghetti. Find me one in metaphase. It's all in a line. All in a line. Yeah, this one. See it? Anaphase, perfect. Find me one that's telophase. That's where you've got 2. There you go. 2, telophase. So sort them into those groups and put them in order. What do you think? Yeah, so you can use this as a guide. Which one looks like a bowl of spaghetti? Yes. No. It's okay. It looks right. What are these?
Oh, anaphase.
Anaphase, what are these? What are these?
Telophase.
Yeah, telophase. So what's the order? What goes first? Okay, interphase. Looks like all of those. Interphase, right? There's a lot of interphase. Then what? What's that called?
Prophase.
Prophase. Then?
Metaphase.
Okay.
Telophase.
Beautiful. You've got it arranged. Now you're going to go through the lab manual, answer this first, and then fill out this table based on what you got.
Why is looking at cells from an array instead of the way to study my
Because it shows the process of how cells
What's the end goal of mitosis?
to replicate the DNA.
To divide and get a new replicated cell. So we can see that by looking at these. What do you guys got? What do you think these are in? Which phase do you think these guys are in?
Interphase.
Interphase, beautiful. What is that? Alright, what else you got? And you can use this as a guide.
Yeah, that's what
Which
we're
one of
doing.
these looks like prophase? This
This
one's
one.
prophase.
Oh wait, no. Yeah.
Kind of looks like spaghetti, yeah, that's a good one. That's a good one.
Spaghetti looking.
Those chromosomes start to condense and you can kind of see the really dark lines.
But they're like,
That would be prophase.
but they're like so dark that you can't even see.
I know, it's hard to see. I wish these weren't in black and white. Most of them, right? Interphase.
But I don't know if you can tell
Last time.
between interface and metaphase because they both look the same.
Well, see, he's got some metaphase ones. Here's an interface. Here's a metaphase. Can you see how they're all lined up on the plate? So that's the main difference. Can you find some anaphase ones? What are those? And what should telophase look like?
Phase, 2 dots.
What have you guys got?
We have most of this.
Okay, what's that?
The intern.
Beautiful. Interphase. Where's your prophase? Prophase. How about metaphase? This guy? Prophase to me. Alright. Where's anaphase? Beautiful. Boom over there. So you got them in order. And telophase. Excellent. So now you have all of your data. You're going to use these to fill this out. What does this look like to you? Give me a descriptive, like what?
The one that's
Yeah, it's definitely anaphase, but like, if you were trying to give me a real world example, like what does this remind you of?
Stick string.
Stick string?
No, the strings that you.
Oh, silly string? Okay. Anything else?
It is like.
Okay, it's like a mirror image. Oh, okay, it looks like a spider. Maybe 2 spiders. Like doing a dance. Okay, teeth. That's the kind of stuff that you want in the descriptive name. You want to give me some sort of descriptive title that will help you remember. Anaphase looks like fighting spiders.
Or I don't know.
Wait, you're saying photo. I mean, that one again. Prophase. Pro. There you go, prophase. Which goes first? Interphase. What's next? Pro. What's next? Excellent. So now you've got this one. Does it make sense? Remember when we watched the video that a lot of cells, most of the time, are in this phase? Remember the pie chart? Like 90% of them are in interphase, and then about 10% are undergoing mitosis. So it makes sense that there's a lot of these cells. Good. So use these data to answer these.
They're egg balls.
They're what?
They're egg balls.
Yeah, either one of those works. Somebody said mold. Alright, what's this?
What about prophase?
Did it break? So let's look at this chart here. Interphase, what's the difference between interphase and prophase? Based on how they look. So the interphase cells, it's like a dark mass. Whereas like she said for prophase, they're like, you can actually see them. So these all look like interphase to me. Doesn't this look like this? Yeah, so let's sort this out. We got some distinction here. Maybe that's prophase. Sort those out. Oh, there's some distinction. I know there are a lot. There we go. How's that fun? Much better. That makes more sense. Do you remember the video that we watched? 90% of the time cells are in interphase. That pie chart, about 90% of them are in interphase. And then the rest of the 10%, they're undergoing one chart. So that's why there's so many interphase. Awesome. Use these data. Fill out this chart. Are you filling out your chart? Garbage, like you can't really see what it says. No, that's self-based. Yeah, start filling this out. You should be done with that chart by the end of class.
Okay. Okay. And phase.
How you guys doing?
T.
Okay, then we have
Like he said.
these are all interphase, correct? What else you got? What's that? Beautiful. Kinesis. Good, really good try. This is the same as this.
Anaphase.
Where do you think it belongs?
Anaphase.
Okay, anaphase. What's this? What are those guys?
Metaphase.
Metaphase. Beautiful. So you've got your piles, so you're doing well.
What about
Interphase.
interphase?
Oopsies. Interphase. This is tough. Let's put them in order so you got some room to think here. So we've got interphase. What goes next? Prophase. Then?
Metaphase.
Metaphase.
Anaphase.
Anaphase.
You got telophase.
Beautiful. Now you're going to use these data to fill out this chart here. You're going to use a descriptive name up here. What does this remind you of? What does this make you think of? A what?
Tearing apart.
Okay, yes, definitely. Something tearing apart, but like, think of like a real world example to help you remember this. This looks like that. If you were describing this to like
Play-Doh.
a little kid.
Play-Doh.
That looks like Play-Doh?
Really? Tear apart.
Okay, tearing apart Play-Doh. Some people have said things like tarantulas fighting. Or fireworks or teeth. Like,
That's messed up too.
so use a descriptive name for each of these in this chart. But I want this chart to be done by the end of class. So I will also This group has about half their chart filled up. Catch up. Are you done? This group has their chart almost done.
We need the percentage.
Okay, wait. You're supposed to be counting up the number of cells in interphase. Yeah. Oh, count up that. That's okay. Most groups have been making that mistake all day today. It's not very clear. Total number of cells in interphase. Put that there. Yes. It's supposed to be a descriptive name. Like this looks like this. This looks like spaghetti. This looks like individual. Yeah, spaghetti. And then the number of cells, you're supposed to be adding that stuff. Is that what you're doing?
Oh, you
Awesome.
mean when we were supposed to write the descriptive names like this, like for prayer, meat, or no?
Adding up.
Yeti.
Just so you know, make a number of cells. You're supposed to be recording the number in each of those cases. Sorry. Make sense?
How many did you get?
Yes. Yes.
Alright, so there's 3 of these.
Those are the actual fibers. So this doesn't happen like magically.
Come on, 37.
The cells all have these things that are called spindle fibers, and they attach to the chromosomes and they yank them with
Prophase.
So that's what that's representing. There's little kind of invisible. We didn't see them in the video. So it's like tiny little invisible strings that grab onto the chromosomes and yank them apart. Yeah.
Telophase.
For the number of cells, you're going to be adding up, counting how many are in interphase, how many are meta, how many are interphase. So everybody can grab a pile.
Oh, I already found that one. It's 12, 6, 7, 44.
What is the next step?
Pictures.
Draw a circle.
Yeah, of course, you can use your calculator.
Who's gonna do pictures?
Have you counted them up yet?
Yeah, 44, 7, 6, 12.
Alright. Perfect. 5 minutes until the exit ticket.
Where's the middle phase? Hey, where's the, oh, they got scattered. That's why. These are there's like 3 middle phase.
Which one is this?
7? And you can use this as a guide. So yeah, I would get those numbers down before you play.
So we have to look at how to make the
That is 12.
It's coming on Wednesday.
He has a couple cousins over there in Afghanistan.
I'm missing something. 1, 2, 3.
Yeah, so now just add these up to get the total number. Divide 55 by the total.
They like playing with potatoes.
Yeah, absolutely.
And they like making farmers. Those are people that drop the pick it up. Get it.
Look, you combine them. What's this? It's okay, quick to fix. How many metaphase do you have? Go. How many prophase do you have? 9.
So we gotta switch around the papers. So here, I'll clean that up.
Ooh, this page is on number, what's it? Page 2 already. Miss, what do you think? We don't need to, but I can count the total. You can write your total over here and then do 44. And that'll be calculator. Yeah, you should calculate. Oh, this page is on page 2 as well.
This page.
But this group, my bad.
This page is on page 2.
So that would be this. This is supposed to be like a descriptive name. Like, see, like 2 is Hey, like, no hands. I don't know. Whatever, just whatever to help you remember this. So that when you're on the test, you're like, oh, choose spider fighting. I know that's anyways.
Yes.
69.
This group is on page 2.
Which phase will be? That's who we'll start now.
My turn, sir.
This group is on page 2.
Alright, which phase is still?
2 more
Do you
minutes.
think it's the longest? Oh, she told us it was the. No, she told us it was the prophase.
Why?
Because look, it says largest phase. The chromosomes are becoming trying to become invisible. They're not invisible. They're becoming invisible. The nuclear memory is breaking down. Basically, it's preparing to separate.
No, stop. What's your question?
It was hard to tell.
The bin.
The other one was dark and the other one was light.
Exactly. Exactly. They're both a circle, but this one has more distinction. Beautiful.
Why do you think cells spend the most time in that phase?
Because they're those host cells. Actually, you know what? Since I already did that. Why do you think cells spend the most time in the phase? No, why do you think cells...
Why do you think they spend the most time?
It's going back to 2. trying to put it down. Yeah, so we can.
Speak your mind.
That's 6 more minutes.
101.
Oh, this group is on page 2. It's a little thing.
She's taking video of her pet.
Yeah. Yeah, it's on page 2.
I like it polka dots.
How am I?
The make. You get polka dotted. Oh. Alright, the lab was an onion.
Alright, at this time, I need your attention. Eyes on me, pencils down. Thanks for tracking. Those of you who are, excellent. Here's what I need. I need you to go ahead and hang on to that lab report. We are going to finish this tomorrow. And find in your bins, you each should have an exit ticket. I want you to complete the exit ticket and put it in the back where we always do. Do that silently and independently. You may see something like this again on your common assessment next week. When you're done, make sure your name is on it and put it over there. When you are done, go back to your seat. You may continue silently working on the lab, but do not talk until everyone is done. Thank you for staying silent. All right, you may commence working on those until the end of class, and you can talk now that everyone's done. Go ahead.
Just interference. Is my turn? Huh? It's fine. I'll put 2 in this. So interface is part of the cell cycle.
Make sure you're trading papers.
Who do you think I'll spend the most time?
We're gonna work on this.
Or do you want
We have 2 more minutes. Sometimes we can get people off.
Sorry.
Wait, it's coming. I can take video of this.
Oh, I'll take your 2 first, but save this. That's good for our review.
I have my gear.
Yeah.
Snapchat.
You can start packing up. Please put beautiful scripts on page 3. Put your chairs on the desk. Yeah.